Okay. Hello, everybody. Again, welcome back to the Atheronet Cardioscope webinar series. Today, we have a pleasure of having Eva with us. She will give a very interesting presentation titled The Impact of Academic Research on Pharmaceutical Success, Insight from Developing Cardiovascular Medicines. Eva is currently Chief Scientist in Dyslipidemia and Atherosclerosis at AstraZeneca, Translational Science and Experimental Medicine, Early Development Cardiovascular, Renal and Metabolism, BioPharmaceutical R&D, and she has been scientist with 25 years of industry experiences in AstraZeneca. And if I'm not mistaken, she's also having a position at Karolinska Institutet. So I think we have a perfect combination of academic and industry experience. And Eva will probably give us very nice overview how we can all advance a little bit more into the kind of industry private part of the drug development story. So Eva, please, thank you so much for joining us today, and you can present everything you have, and then after that we will start with the discussion session.
Thank you so much. Thank you so much, Miro, and everybody else who organized these meetings. It's a very nice idea, and it's a pleasure for me really to be here. The reason why I'm here is because Miro listened to me in a presentation recently in the University of Belgrade, and he found that this is something that the audience of the Atheronet could be very much interested in. When I give this kind of lecture or seminar, I'm thinking in particularly in young people, in young scientists. When I work at Karolinska Institute, many young scientists, PhD students, postdocs approach me and ask me about the differences working in the industry versus working in academia. What shall they do? Shall they move to here and there? They are afraid of leaving the science. They think that moving into industry means that no more science. And I just want to tell you, and this is one of my first message, that is not true. That depends on you. If you want to continue being a scientist, of course, there are plenty of opportunities in the pharma industry to work as a scientist and to continue working as a scientist. So I hope that I can share with you some of these ideas and experience, and that's why the title of this presentation, I also wrote some personal reflections. So do you see the next slide?
Yes.
Yes, it works okay.
So a little bit of the content so that you know what we are facing in the coming 40 minutes. I will introduce myself, my career, again, with the thinking that hopefully for young scientists and for young scientists, it's an idea of how can you do career moving from the academia to pharmaceutical industry. And then I will go into the process of drug discovery. What does it mean to develop a new drug? There are essential steps that are applicable to any disease area. And I work mostly in dyslipidemia and atherosclerosis cardiovascular disease, but I'm going to present the process of modern pharmaceutical company that actually are, again, applicable to any other therapy area. Then an example of how one of the more modern medicines that are still in clinical development, some of them already available as monoclonal antibodies, but there are others coming behind, how that was discovered purely in a scientific environment and not in the industry, but how the industry immediately saw the opportunity of this science to be applicable to develop a new medicine for the treatment of hypercholesterolemia and aiming for reducing cardiovascular events. And then at the end, I will share, after 25 years of experience working in AstraZeneca, I will share with you some personal reflections as a scientist working in the industry. Important also to say is that the content of this presentation does not necessarily reflect the opinion of AstraZeneca. I got permission to show all the slides that you will see, but I am in my comments, I am independent on what AstraZeneca opinion, and that's good for you because that means I am not here doing any propaganda or advertisement for a specific company. I am here talking as a scientist. When it comes to career, I started my career do you see if I point the slides?
Yes, we can see. Yes.
Great. I started my career in Caracas, Venezuela, where I studied biology at the university, and then I did a master's degree in biochemistry. And then I moved to the faculty to Gothenburg, Sweden, in 1992. I did my PhD. And the reasons for moving to Sweden were unfortunately due to economical and political situation in the country, where I didn't see there were going to be many opportunities for me to develop as a scientist and do it as a career. So I got the opportunity to move to Sweden, and I did my PhD at the Faculty of Medicine. And then immediately I continued the classical career at the university, where I got the associate, became associate professor. Eventually, I became the director of the Wallenberg Laboratory for Cardiovascular Research, and this was an extraordinary experience from the administrative and leadership point of view. I was appointed later as professor in cardiovascular research, and also simultaneously, almost at the same time, I got the invitation from AstraZeneca in the year 2000 to move here to AstraZeneca and to work with AstraZeneca, establishing the research area in the atherosclerosis cardiovascular space. The first thing that I did when I moved to AstraZeneca after 15 years in the academia, you understand, I'm sure, it was not easy for me to leave back 15 years of successful actually academic research and move fully to the industry. So I requested that, yes, I think that moving to the industry is very interesting, and that I could contribute with my knowledge of the disease to drug development. But I wanted to keep a foot outside. I wanted to keep part of me outside in the academia still. It was good that AstraZeneca at that time, and still today here in Sweden, they are in that sense quite flexible. And yes, it is possible to combine your full-time employee in AstraZeneca with a 20% time outside in an institute that is, of course, well known, should be well known, and that that there is a team where you are associated with, and that there is a clear objective and with clear goals to achieve every year. And since then, I have been doing that, and I have been actually combining my role in AstraZeneca with other adjuvant professors' positions from Linköping, Gothenburg, and also in Karolinska. Nowadays, I am in Karolinska Institute in Solna, in the translational science, in the translational medicine surgery group. And when it comes to AstraZeneca, I had many management administrative positions, from team leader to head of the department. But then it came a moment in which I decided I want to work as a scientist. And with that in mind, I decided I'm going to proceed with a scientific career inside the company. And I started with that since the beginning, because I always was attached to science and publishing. And then I started to get involved in research activities, such as establishing contacts in Japan with investigator research in Japan, in order to identify ideas that could be of interest for us for developing new medicines. And also I work at time as a director in the group that deals with externalization, and in order to learn more about what is needed in terms of paperwork and in terms of confidentiality and non-confidentiality aspect when you work with external groups. And then I also internally in AstraZeneca AstraZeneca 2017 decided to have their own global postdoc program. So we have every year coming to AstraZeneca postdocs that work here in the company, and I was involved very much in the postdoc committee. And nowadays, I left that after many years working on that. And finally, I reached what it's the highest position that you can have as a scientist, purely scientist in the pharma industry, that is to become chief scientist in a specific disease area. So this is just to give you, this is not about talking about me. This is just to provide an example for young scientists. If you want to do a career in a pharmaceutical industry, of course you can do it. It depends on you. And of course you should bring up this inside the company with your colleagues and new line managers and discuss the opportunities. But it is possible. The first thing that you need to think about if you want to develop a new drug, a new medicine, is if there is a medical need. There need to be a medical need that justify the work involved in developing a new medicine. That's the first question. So if you have a program in your research, you have a fantastic finding that could be applicable to develop a new drug, a new medicine, think about it. Okay, it is new, but is it needed in the society? And in that, this slide shows that in terms of the cardiovascular disease, there is still opening for new medicines because it is the major global health problem. And this slide, although the title is global health, the slides to the left, the bars are showing that the cardiovascular disease in the United States is appearing nowadays in early age. You can see here, in men and women, the presence of atherosclerosis cardiovascular disease actually is starting to appear already in the 30s, 39 years of age in both genders. And then if we move to the right here, you have the Europe map. And in Europe, of course, the incidence of cardiovascular disease in the different countries. And there is no doubt that the cardiovascular disease is still a main health problem in Europe. And attached to that, of course, there is a cost. And below here is the different risk factors that are associated with the development of cardiovascular disease. And although we do have medicines today to control blood pressure, to control high lipids levels, and so on, interestingly, in the area of high lipids level, high cholesterol, there is still today a main risk factor for the development of cardiovascular disease, telling us that despite the availability of lipid-lowering drugs such as statin, ezetimibe, etc., there is still a window for a need for new medicines because it's still a problem in the society. What is the definition of a therapeutic drug target? A drug target is a molecule in the body, usually a protein or a DNA or RNA, that is associated with a particular disease process and that could be addressed by a drug to produce a desired therapeutic effect. And this is something that had happened and has expanded in the last 20 years. Two things happen. One is that now we talk about proteins and DNA and RNA. And as a matter of fact, I realized I should modify this slide and I should include also cells. But also there is another thing that had happened, and is the concept of what a drug is. And today, in the last 20 years, there have been so much development that we have many different modalities as shown in this slide. You can see here that when I joined AstraZeneca 25 years ago, this was what we were working, small molecules, small chemical molecules. In the cardiovascular area, that was the only thing that was available. Yes, there were some antibody therapies, in possibly mostly in cancer and in inflammation, and there were some peptides or proteins like insulin, but in general, the focus was small molecules. But today, as you can see, there are many possibilities to develop new medicine with antibodies, with peptides, with proteins, with cell therapeutics, and very much with nucleotide antisense molecules, silencing RNA that you can reach the gene expression of a particular protein. And this, of course, has made, as one of our previous vice presidents in AstraZeneca said, that actually there is no limit, there is no target that we cannot achieve, that we cannot reach, because there are so many modalities we can use in order to modify something that is involved in the development of a disease. But there is also another thing that is attached to this expansion of the portfolio of modern pharma industry with the different modalities. And it is the fact that you also require ways, mechanisms, solutions to deliver those medicines to the tissues. Because a small molecule, a chemical compound, it's a small molecule. Here you can see an example of the molecular size, and you can dissolve it in a formulation in a form that the intestine can absorb it, and it comes inside the body and is exposed to the tissues where the molecule is expected to exert an effect. But when you start to work with other modalities, such as antibodies, such as oligonucleotides, silencing RNA, then you cannot provide administer this drug just by the intestine, because it will not be absorbed by the intestine. Now you need to inject them either intravenous or subcutaneous, and also they are very much water soluble and they will dilute in the volume of the body. So you need to provide them in a formulation that many times is about nanoparticles, in order for the drug to achieve, or the molecule to achieve, the organ where it is supposed to exert an action. And also, once reach that organ, we also need to see if we can use specific receptors that are expressed, for example, in heart cells or in liver cells, and that receptor has a ligand in the nanoparticle that is being used to transport the new drug. So there has been an expansion in the modalities, and at the same time, there has been, as a consequence of that, there has been an expansion in the other part of pharma industry that is responsible for the formulation, how to administer the medicine, these new medicines to the patients. In pharma industry, independently where you are, in which therapy area, when you move into, the first thing is that you have a preclinical package. You have research, you have in vitro data, you have in vivo data, you have a hypothesis, and you can show that hypothesis, you can modulate that hypothesis in vitro and in vivo, and it has an effect. And now you are prepared to move into clinic. And in clinic, we have usually a phase 1, a phase 2, a phase 3 periods. And in that, we have, the objectives are different. In phase 1, is mostly about safety and tolerability. In phase 2, you start now to dose in different concentrations of the medicine in order to see the efficacy. In phase 3, now you want to see whether the drug behaves better than the gold standard that is being used currently. And then the phase 4, of course, now is at the post-marketing, and now you have a product, and now now the patients can achieve the new medicine. But these steps are, all of them are are used daily in the process of finding, developing a new medicine. How much does it cost to develop a new drug? Well, it's very expensive. It's about $2.6 billion, American dollars. But why is such? Because in the cost of a new medicine that made it to the market, you also need to include all the failures. So failures are very expensive for the pharma industry because you need to add that cost to the cost of a new drug. You cannot recover anything that you have invested in that failure. So that's one of the reasons why the cost of a new medicine is so high. And also very few, less than 10% of the drugs that are tested in the phase 1 period of the clinical drug development actually move ahead and are approved. And also the majority of the failures are due to two things. One is the lack of efficacy, and the other, of course, is safety and toxicology. Both are extremely important. You need to see that you are achieving a change in the body in vivo that will be associated with a benefit in terms of the disease. And of course, the medicines should also be safe in order to be used. So in selecting the right target, the right mechanism for developing a new medicine is a very important decision in this process. We in AstraZeneca, we use something that is called the 5 R's, and we need always to think about the right target we want to identify. And here is a lot about validation, scientific validation. We need to find also the right tissue, which is the tissue we want to approach mostly with our new medicine, and how to follow the effect in that tissue. Of course, we need to look that the new molecule is safe, very important. We also need to think about the patients. I mean, sometimes you want to have you develop a new medicine for a specific clinical phenotype, or you sometimes want to have something that is more broader, many more people can benefit of that. You need to have that idea already from the beginning. And look, if you are selecting specific patients, you need to have ways how to identify those patients. For example, you need to have a special biomarkers in plasma. There is a protein specifically that is elevated. You need to have methods that are clinically approved to measure that protein and that is associated with a disease. And then, of course, not last but not least, of course, in the private pharma industry, there need to be a right commercial case. I mean, as I say at the beginning, there need to be a medical need to develop a new medicine in order that we can sell that new medicine, and the cost that has been invested in developing a new drug can be paid, including, of course, salary of the people. The drug discovery, again, when you select a new molecule, what we call a new candidate drug, you can imagine that it is about like starting with a lot of stones, and then you start to select the best stones, and then after the best stones, you find finally the jewelry. And this process is very time consuming. And also you need to leave many stones because, yes, you need to find a molecule that will be safe. Not only that is doing what you want the molecule to do, but that you guarantee that during the treatment use, the drug will not harm the patient. Human target validation, of course, as I said before, is very important. It's the first step. It's a really, really first step. You in the academia is classical, and you will see an example of that. You find out something, you find a new mutation that is associated either with development of the disease or with protection of a disease. You want now to develop that, to proceed and use that idea, that finding for developing a new medicine. This is the human target validation. It's the first step, and it needs to be associated with a clinical benefit. And there are many types of scientific evidence to contribute to human target validation. One of them is the genetics. The other is the multiomics, transcriptomics, proteomics. Then, of course, you have the population data, epidemiology, and of course, sometimes you already have intervention studies with the same, using drugs with a similar mechanism of action, but you are now moving into a better modality. So, but that tells you that if the previous intervention studies have been successful, of course, it brings more, increases the rate of success of a new drug. The genetics has become has a high impact in the development of new medicine in the last years, and in the fact that we have expanded into new therapeutic modalities. For example, the classical one are the antisense molecules, the silencing RNA molecules. Why? Because having an antisense or a silencing RNA molecule means you want to reach the nucleus of the cell, and you want to delete, or you want you want to stop the translation of the mRNA into a protein. Sometimes even you want to stop the transcription of the DNA to the mRNA in order to stop the production of the protein that you want. So, and for that, finding loss of function mutations that are associated with a positive clinical phenotype tells you that if I inhibit that gene, a particular gene, if I inhibit expression of that particular gene, I may mimic the phenotype that I see in humans that are carriers of loss of function mutations in a specific gene. And there are reasons why the genetics make a good genetics make a good target, and is that, of course, genes underlying monogenic diseases are a good starting point because they have extreme phenotypes, but that are associated with a high penetrance. It means there are few people carrying the loss of function mutation gene. It's a rare gene, but if I mimic it, I can expand it to a broader patient population. The Mendelian disorders can support mechanism of action for a gene or a pathway. We can do Mendelian randomization studies in population to support the genetics. The majority, a high percentage of Mendelian genes are located near the loci of common diseases, and not less, the genetics can be copied in preclinical in vitro and in vivo models. You can do loss of function mutations in mice or in cells, or the opposite, you can overexpress. So genetics has had a tremendous contribution to the last year's expansion in the modalities for new medicines. Human target validation, why it is important, as I say before. This slide shows you results from an internal AstraZeneca evaluation that was published some years ago in Nature Review. And there are two papers, one in 2014 and the other one came in 2018, where using internal data from AstraZeneca and also available external data, they evaluated in here what are the reasons why the projects fail and what are the reasons why the projects succeed. And when they finished this study, it was clear that the majority of the projects failed in what is called the phase 2 that you saw at the beginning of my presentation. Is here the majority of the clinical trials are stopped and they don't proceed. And in phase 2, if you remember the first slide, what you do here is that you start to look at the efficacy of the drug, meaning the drug is, you want to see that the drug is doing what you expect the drug to do. But then when you test that drug in humans, you realize that the drug is not doing that. So that means that the hypothesis you had from the beginning is not correct or was not correct. Also, the project closers are why you close a project. It's not only, yes, it is, sometimes you can close a project because the company changed their organizations, changed the strategy. That's what could be one reason. But again, the majority of the project that fails are due in, as shown here, is in phase 2 and is due to the lack of clinical efficacy. And then, of course, that means they also look at why the reason for the lack of clinical efficacy. And then you start to look into the preclinical data and you start maybe to find out gaps in the translation between animal models and humans, or between experiments performed in human cells or in animal cells into humans. So it's a very much about the preclinical information that we had accumulated at the beginning before going into clinic. So the problem starts here in preclinical. Of course, drug safety is also a big problem. And also in this AstraZeneca internal evaluation, it was clear that many projects already from the beginning in preclinical are closed because of the safety. I mean, that's the real life, and you cannot do anything else than stop the process. That doesn't mean that necessarily the safety is due to the target. The safety can be related to the drug, the small molecule, or the antisense, or is associated with the chemistry. But sometimes it can also be associated to the target. After this internal evaluation that AstraZeneca performed, as well as many other companies, safety was something that many years back traditionally was performed late in development. But nowadays, safety starts from the beginning. Since the discussion of the idea of the hypothesis, safety is involved. And that has shown to produce a pipeline with a very low attrition rate due to safety problems. Another classical question I get is where do the ideas to develop new medicines come from? And this slide shows you real data from us, from AstraZeneca, and from the year 2018 to the year 2024. You can see that the majority of the projects that we are working with comes from the scientific literature. No way. You need to follow the literature, you need to read, you need to understand what's happening in order to identify new opportunities. And here we are going back to the question about the importance of having scientists inside the company working in developing new medicines, because you need to follow what's going on in the academia, what's going on, what is new. Otherwise, you will not reach that new information. Of course, we have also in licensing with external groups. That's also happened. As I say before, you have the human genomics. That is something that is more new. And you have also sometimes human omics. So I will say scientific literature and in-house human genomic information. This is an example of what happened, as I said before, of the most recent medicine for the treatment of hypercholesterolemia associated with cardiovascular disease, myocardial infarction, as an example of how the science is the basis for the development of new drugs. And here you have an individual, a man that came in the hospital in Paris in 1999 with a massive infarct. And this gentleman survived a massive infarct, and something that you do normally today in the hospitals, if somebody has an infarct, is that you take the blood samples, you measure the cholesterol, and if the cholesterol is very high, they send samples to the clinical lab to do the classical, genetic scoring. They look for classical genetic mutations that are associated to hypercholesterolemia, such as mutations in the LDL receptor, the low density lipoprotein receptor in the liver. And they also look at other mutations that are known to be associated with hypercholesterolemia. But in this case, this person didn't have any mutations in what was known at that time, 1999, that is associated with developing hypercholesterolemia. So why this person has such high level of cholesterol? So the clinical genetic group in that hospital in Paris appointed a young fellow, Marianne Abifadel, to sequence the DNA of this patient and the family and find out what's happening. And Marianne identified a new gene, a mutation in one gene that was a gain of function mutation. And that gene has a terrible name, that is PCSK9. And nobody that was never reported, never in life, what PCSK9 is doing. So Marianne and the team in Paris went to Montreal to work with a person, Professor Nabil Said, that is expert in PCSK9 proteins, but he himself never worked with that lipoproteins and lipids and cholesterol. But they started to do the classical in vitro experiment expressing the PCSK9 mutation that they have identified. And so morning on, all the people started to do animal models, also mimicking the mutation. And everybody find out that, yes, if you carry a mutation in the PCSK9 that make that protein non-functional, you develop hypercholesterolemia. And later on came another group in Dallas that had access to human data, and they started to look for the mutations and other mutations in the PCSK9 in population data associated both with very high level of cholesterol or with low level of cholesterol. And they could confirm that the mutation of this person that appeared in Paris was a mutation that is found also in the United States in population studies, but also the opposite, that you can have mutations that lead to non-function of the protein or non-expression of the protein, and those are associated with low cholesterol level or lower cholesterol level and low cardiovascular risk. So all this is the basis for the new medicine being developed in Novartis, in AstraZeneca, in Merck, in Amgen. All the main big companies are working with this concept and developing and have developed efficacious monoclonal antibodies to block the PCSK9 and to reduce cardiovascular disease. So it's an example of, without the work of these scientists, there wouldn't have been any new medicine. So science is really important, and following what's happening in science is even more important. This is what had happened since 1999 with the discovery of or 2003 when the gain of function mutation was published, and all the new drugs that are being developed in the pharmaceutical companies. Of course, cardiovascular disease is, cardiovascular disease is not just a monogenetic disease. Monogenetic causes like deficiency in LDL receptor and PCSK9 overexpression can contribute to the hypercholesterolemia, but we know that there are many other factors that contribute to atherosclerosis cardiovascular disease, such as remnant lipoproteins, another lipoprotein that is in fashion nowadays, that is little lipoprotein A, the metabolic pattern profile of the people, of course, inflammation in the arteries, and not less, of course, thrombosis risk. So, and this, we are not going through that. This slide for me is just to tell you that there are nowadays many other medicines that are being developed in other pathways, in other dealing with other mechanisms of action, but all of them are really based on science, on information that has been developed, that has been generated in the laboratories. So now the last 5 minutes I want to use to share some personal reflections. I finished now with the drug discovery part. I hope that you find it useful, that you understand now that science, data, preclinical data is essential for developing a new medicine, and that preclinical data need to be translated in humans. You need to find is it how if it is associated with disease risk or with disease prevention in humans, and then also the question, how shall we target? Shall we use a small molecule, an antisense, a monoclonal antibody, and then you start to build the package. But now I want to share with you what is experience working as a scientist in a global corporate with more than 65,000 employees. Well, number one, you are a scientist, and that's what is important. You have a great educational training. You have a PhD, sometimes 2 postdocs. You have already a background of education that allows you to find information, to analyze information, and to make conclusions. If you can also, you know also how and where to find information if you need it. So you have already a very good training, even if you will not end up doing something else that was not that you were not thinking about when you were doing your PhD. What is important is that you have the education with you. Then in a global corporate, there are different roles. You can work, you can have an ambition to become a leader, a CEO, a vice president, a director, a head of the department. You can have, you want to be a project leader, you want to be close to developing the idea into a new medicine, or you can continue, you want to work in the laboratory. You are a wet lab person in cell biology, molecular biology, veterinary, biochemistry, and I didn't put here much very important chemistry. So you have different alternatives for different roles in a pharmaceutical company. What you need to think about also is if you want to work, if you want to develop your career into in leadership, think about that the skill needed is communication. If you want to become a project leader, the critical skill to have is you need to be a very structured person. And of course, if you want to keep working in the wet lab, of course, what moves all scientists is curiosity. So this is just that there are different critical skills that characterize different group of roles. Also, with the progress and the career developments, I think that you should maintain and build your scientific, technique, or business competence. Anything that you have in your shoulder, in your bag, that brings you into a large company, you should keep it alive. You should maintain it and or build it. And also remember that you are part of an industrial team. That means you cannot be individual. At the same time, it's good that you keep yourself, your individual profile that make you unique or different. This is very important if you want to develop in a particular role, because if you don't do that, you become one more in a large industry. And also think about it that compromise is the secret in order to be part of the future in a global corporate. I mean, there are many opinions, many experience. Many times you will find yourself that you don't agree with what other people is saying or doing or the decisions that are being taken, but compromise is part of surviving in a large global corporate, because there are, again, the variety is and of people and the variety of situations is enormous. Think about it. There are big differences between academia and industry. When you are in the academia, it's about you, the individual. You give the Nobel Prize to one person. You don't give the Nobel Prize to a team. So academia is extremely individualistic. It's about your career, your idea, your publication, the first author or the last author. In the industry, it's about a team. You are part of a team. In academia, your idea, your hypothesis, now that idea became the company-owned idea. And you need to recognize that. One discipline is about your knowledge. Many times even working with other colleagues that have the same knowledge, molecular biology or cell biology or animal models. But in the industry, it's very much multidisciplinary from the beginning. Different disciplines are needed in order to validate an idea, because now it's not any longer about one hypothesis. It's about how that hypothesis will impact a broader number of patients. Personal merits in the academia, of course, in industry is about business focus. It's about investment. And of course, in academia is your intellectual achievement, but in the industry is about launching a new novel medicine. And last but not least, be positive. Progress doesn't have time for negative people. I can tell you that every day. Accept and agree are not the same thing. You accept reality, but you don't necessarily agree. It's also very important. It's about the compromise. And last but not least, learn from everything that you do and measure it. Ask yourself every day, what I have learned today. You need to keep growing all the time. And with that, I finish. I don't know if we have time for questions, but I hope you find this in one way useful. Thank you so much for your attention.
Thank you so much for giving this wonderful presentation. Also, before it's the day before the holidays, I think it's good to have this inspirational talk to give us a little bit time to kind of process it during the holidays. So I really, really thank you so much for giving us the insights how drug development is happening right now, how the academia is part of this process, and I think we really can start with a discussion regarding this topic. I opened this webinar for discussion. We have questions on the people in the audience. I will just request to unmute you, and then you need to accept this request. Yeah, thank you. Luis Perpetuo, sorry.
Close enough.
Yes, thank you.
Can you hear me?
Yes.
Yeah, okay. So thank you for everyone organizing this meeting, and thank you, Eva, for making this lovely presentation. I am a PhD student, right? And I often contemplate if it would be better to, not better, but contemplate the options of going through academia or going into the industry. And I try to make the best question I could. So personally for you, what gives you or what gave you more satisfaction, to work in the academia or to work in the industry? And if whatever you choose, what gave you that satisfaction?
You are muted, Eva.
Sorry, thank you so much. Important question, of course. I mean, of course, you need to choose what you make you more happy. I did have a very good scientific career in the hospital, but I got the opportunity. I mean, they knocked the door and asked me, "Do you want to move to AstraZeneca and to do this?" And somehow, for me, that I'm sharing with you my own experience, I thought I mean, I didn't think about that before, but when I got the question, it was like opening a window and I started to see the possibilities. Just for the change, I found it interesting. I say yes. Always, I say, if it doesn't work, I can always go back to the university. And it had happened. I know people who had joined the companies and they moved back to the academy, to the pharma, to the academia. And then what made me, I mean, 25 years in AstraZeneca, 20 more or less, 15 in academia. It gave me, today I can tell you that I have a lot of satisfaction working here in AstraZeneca. But of course, I cannot deny that there have also been very difficult times because it's large companies, no, and they can't be taking decisions that are beyond your wish. But as long as you enjoy and as long as you feel that you are doing something useful, I think that that is the motor that drives your decision.
Thank you. We have another question from the audience. Let me just see. Was the next one? It was
Luis Masana.
yes, yes. I'm sending the request to unmute.
Okay, thank you.
Yeah.
Hi, Eva, how are you? It's Luis.
Is it Luis?
Yes, it's me.
Coño, Luis, ¿tú estás escuchando esto?
Of course I was.
Sorry, I'm speaking Spanish with Luis because I know him. Well, good. Good to know that you're here.
Well, it was a brilliant presentation as usual. I have a couple of questions for you. One of them is, in the last time we've seen that the kind of drug development moves to from small molecules given orally once a day, several times a day, etc., to subcutaneous therapies.
Yeah.
Probably because the molecules are more complex because they're proteins, etc. But I see now a kind of reverse pathway. So
Yeah.
Astra, for example, is developing again
Yeah.
molecules that will act in very well-known targets that
Yeah.
that are now treated with subcutaneous therapies, and now will be also again done by or treated by orally
Yeah.
therapies. What do you think
Yeah.
is the future of this kind of
Yeah. I
therapy?
know where you are going, and that's true. That's true. I think that it's a combination, Luis. The reason for that was the failure of the PCSK9 monoclonal antibodies to reach the market. They started with very high prices, as you know, and regulate. And because there are very high prices, then they carry with them a bureaucratic procedure in order to select the patient that really can use it. So, and then they started to reduce the price in order to make it more accessible. We cannot underestimate that the way how the two monoclonal antibodies that are available today in the market came into the market actually affected the development and acceptance of other injectable medicines. If they would have had a more successful and more easy way to reach the market, I think that today the scenario would have been different. That said, there is also another component. For some diseases, like in oncology, like in rheumatoid arthritis, inflammatory, you accept more the injectables. It could be because it's for shorter treatment of time, but in the area where you and I work, that is dyslipidemia, atherosclerosis, it's for chronic treatment. Once you start the treatment, you should take it forever. And that means, of course, there you can also think about having an injection once a year. Why not? But that still we have not tested. And if Novartis is successful with their inclisiran, it could be, if they are smart and they have a good strategy how how to penetrate the market, they may change again the concept of injectable versus a small molecule. But still in cardiovascular, with experience with hypertension, with experience with drugs given in diabetes, people prefer the oral molecule.
Okay, thank you very much.
But again, my answer to you is it's not only about the concept of an injection versus a small molecule. It's also how the marketing of that product ended up. A bad marketing can affect the availability of new drugs using the same modality.
Thank you. Yeah. Congratulations for your time.
See you in Glasgow.
See you.
We have a question from panelists. So Yael, you can go, and then Paolo, you can take over, and then we will dedicate some time to the questions from the chat.
Yes, thank you so much, Eva. That was so interesting. I am a researcher in academia, and I have no aspirations to go into industry, but I would like it very much that whatever I discover or research has an application later on. But of course, I'm driven to publish as much as possible, as soon as possible, which means that we very often do not patent our findings and publish them. Can those findings, I mean, I saw the PCSK9 discovery was academic, and then later development is industrial, but can findings that we publish still be developed into drug targets, and how much knowledge do we publish and thereby throw away? Or is that not an issue?
That's a catch-22 question, because number one, in genetics, the finding of the PCSK9 as well as other genetics, you cannot patent, because that's biology. You didn't discover anything. That the mutation was done by nature. So there you cannot patent. Now you have another thing that you can patent, and you have a patent, and then you don't have the publication. But then you enter in this catch-22 that industry also want to have validation, as I say. And the best validation for a new idea, as you know, is that somebody else also find the same as you. Otherwise, it may be that what you did alone is really serendipity. It's nothing, you see. So in that sense, publishing is good, because you put your data in the public domain. That's it. This is what I have, you know. And then other people, and you are the first author. The group is the first author. It's the first publication. Everybody should refer to you. If they don't refer to you, then they are not working ethically. So you need to discuss it with a patent lawyer. Sometimes there are things that you can patent, and then the rest you publish. In AstraZeneca, I can tell you, we are very much prone for publication, even in our internal work. Very much to publish.
Thanks. Thank you very much.
Paolo?
Eva and Paolo, well, we don't know each other. I hope we can meet in Glasgow. And I mean, your webinar was really inspiring. And not only that, but you still have a piece of your heart in the academia, this 20%, if I got it right. Why? So this means that, I mean, one word is not enough for you, but you would like to have some bias also of the other word. I can understand this, because it happened to me a couple of times in my life to maybe have some opportunities. I say, well, let's stay in the academia, but then a piece of something. But you have been more courageous than me, and so probably anyway.
Well, listen, maybe because I already was working 15 years in the academia, and even if I decided to move to the industry, it was like, it was a sentimental thing. I say, I don't want to leave. I don't want to throw away all these years. You see what I mean? I want all these years to be with me and also help me in my career inside the company, because having a network is very important. And I already built a network of scientists, and that's what AstraZeneca wanted. They wanted me to build a new area of research inside the company. And for that, you need to have contact in the academia. You need to know the people. And if I didn't, I wouldn't be, then I become dependent of other people. So, and today, that's something I enjoy tremendously. For me, it's just to call by telephone and talk to you and talk to Miro and talk to others because we know each other. So I don't need a confidential agreement paper in front of me. We have a gentleman agreement. I know what I can talk and you know what you can tell me. So it is, I think that back to your question, for me, I think sincerely, it was a little bit, I didn't want to leave everything. I want that to be part of me. And then I didn't know how much time the company was going to take from me. And again, nothing wrong with following the science. And if I can follow the science by working with an external group one day a week or one day a month, that's great. And do with them I'm not doing inside the company, so that is not duplication.
Great combination. It's really very nice.
But also you need to have companies that allow you for that, no?
Well, looking forward to meet you in Glasgow and putting
Me
together all of us some good things
Me
with
too.
all the colleagues that are present here and some
Me too.
others.
Me
Thank
too.
you very much.
We
Thank
have two more questions from the chat. One is from Stephanie. She's from Malta. She's running a very nice study on acute coronary syndrome, and she has very nice multiomics data. And this is why I guess she asks, how important are human data sets in academia? For example, multiomics data sets for pharmaceutical industry. And what ways would you recommend for the academia to collaborate directly with pharma companies? What would be the best way to interact?
Offer to the company what you have. My experience with many groups dealing with biobanks, proteomics, omics, is that they keep that for themselves. And then years pass, and they have been used very little. So if you have information, omic data, population data, and you think that offer that to a company, tell them I have this, you can use it for this. You know why? Because then many other, if you want to confirm data, then the company can do that. If you provide them with plasma data, or if you can provide them with omic data, then you need to validate your findings in another patient population or run an assay to measure that protein, for example, in plasma. Then the company can do that. So that it's a win-win. It's a win-win. I see so many biobanks dying as dinosaurs because they are not being used. They're afraid because they want to have money. They want, and you cannot do money with biological data. You need to understand that. You see what I mean? Not because you collected the data, you are the owner. Fine for your publications, but that's it. So, recommendation: if you have information that you think is useful for a specific company, for a specific therapy area, contact them.
Great advice. We have another question from a senior researcher from Italy, Fabio Martelli. He thanks you for the presentation and asks how easy it is to go back to academia after mid to longer than 5 years experience in the company.
I don't know, because I haven't done it. But if it's something that you want to do and need to do, of course, it will depend of having an opportunity. Maybe you need to apply to a new grant, you know how. But I mean, if you are motivated because you're not happy in the pharma industry and you go back, of course, you need to look for where you go back and what you need. But I don't see that as something that is impossible to do. And by the contrary, you carry now 5 years of experience working in pharma industry that can be very much applicable when you write a grant. Now you know many details about pharma industry. that you can expand your grants, not only from basic science to something more applicable.
Great, thanks.
I would like to stay here longer, but I can't because I have a meeting and people is waiting for me.
Thank you so much for your time.
Thank
We really
you.
appreciate it. We would end it anyway.
Miron, you can send me the questions and I can answer and send it to you, okay?
Okay. Thank you so much. Thanks, everybody.
you.
See you in Glasgow, whoever is coming, and have a nice holiday tomorrow. Sorry.
The same to you. Thank you so much.
Bye
Thank you so
bye.
much.
Thank you
Thank you
very
very
much.
Bye-bye.
much. Thank you so
Bye.
much.
Thank you.